So yes, my name is Vinicio. I'm an HPP application specialist at Hyperbaric. Any questions related to HPP, food processing, regulations, packaging, I can always give you help. And today we'll be covering a little bit of the basics in terms of the food safety and nutrition. Again, setting up Grace's presentation later for the evening. So we start with a survey. Here a master dissertation here by UNL in which Dr. Grace collaborated or directed. They basically took samples through a whole year from companies that were selling raw pet food and raw pet food items to consumers online. So think it as a worst case because it took a couple of days for samples to arrive. Temperature abuse conditions because shipping, well, it may be not necessarily the best practices in order to move these delicate products. And again, they basically throughout a year they sampled recipes out of beef, chicken, pork, and even though here is not shown, also from turkey. They analyze this in ground meat organs, meat, muscle, tendons. This is the plot on the left. On the right, they also compare this to liver, and we have the enterobacteria counts, which is basically like a gut microorganism indicator, very generic. So over here you can also kind of include Salmonella, generic E. coli, and you can see, well, the counts, the limits over here suggested by the European Union for raw pet feed. Here in the USA there are currently no standards for these quality indicators, but you can see all data points in red indicate that it is above the recommended or the indicated limit, which corresponds to 30 to 40% of the samples analyzed. So again, it's not only the enterobacteria, but the survey also indicated that there's a presence of generic E. coli, generic Salmonella, Listeria, and well, if these organisms are present in greater counts, there's also increases the possibility of having the pathogenic strains of Salmonella, Listeria, which due to regulations should not be present at all. And the same thing applies for pathogenic E. coli. That's where HPP comes into place. As previously mentioned in Tony and the discussion with Grace, pathogens may survive freezing, may survive freeze drying. So you need a lethal step in order to minimize risk with these foodborne pathogens. HPP, a non-thermal technology, you use cold water at very high pressure in order to reduce this risk with bacterial pathogens, viruses, and parasites. It is being long used for the human food industry. It started all the way in the early 1990s in Japan. Here in the US, the boom in the late 1990s, mid-1990s with avocado products, ready-to-eat meats with Listeria loss of high incidences due to contamination during slicing or other maneuvering during packaging. And you can see that, well, juices and beverages take nearly 25% of installed HPP capacity. Meat products, along with the pet food, again, it's the fastest growing segment for the HPP industry, will take another 20%. Dips or plant-based salsas, complex plant-based ready meals take another 20%, and that's a big overview of how HPP is applied currently within the food industry. For pet food, there are two main or major applications. The first one will be when you apply the HPP to the raw blends, meats or the muscle, organ meats, grains, vegetables, dairy, eggs, whatever you need to meet the nutritional requirements of the pets. They are all blended together. They can be sold refrigerated, frozen, or apply HPP as a prior step before the freeze drying. Again, this research group, they basically evaluated 6,000 bars, 87,000 PSI, 3 minutes, which is kind of like the standard recipe or for HPP food applications. Different diets, you can see again it's the muscle meats with the organs, with the veggies, eggshells, dairy, etc. 3 different diets, and over here in the plot, you can see diet number 1 unprocessed, the open blue bars, the closed blue bars correspond to diet 1, and so on for diets 2 and 3. You can see again the changes in terms of the protein content, the fat, the carbohydrates. are basically the same before and after the process. So some companies that are dedicated to this dive into deeper detail. They actually go and analyze individual components of the nutritional properties of foods. You can see over here results by different independent studies of instinct pet foods, Steve's Real Food. Here are the links in case that you want to consult additional information. But again, they measure the content of individual amino acids. You can have a couple over here, the large or the protein content, some healthy fatty acids. And again, if you take a closer look on the x-axis, most of these components, the change is in the -1 to +1 scale. And that basically means, again, it's the same before and after. These small changes may be more associated towards natural variability, instrumental error, rather than an effect of the HPP. It's not perfect. In example, HPP can induce lipid oxidation. Steve's Real Food, in example, reported the loss of vitamin A, but through reformulation, by adding more liver, they were able to counteract this loss induced by the HPP. Is the color change induced to these structural changes from the protein. You can see that it's quite visible, starting to 200 megapascals, which will be around 30,000 psi. And as you increase the pressure level in order to eliminate pathogens, this becomes even more noticeable. It is the redness, it is also the lightness or the perceived opaqueness of the protein, and we will be covering more this in the pilot plan demonstration. This is also the color change, particularly the redness related to oxygen exposure. As you can see, well, some research studies by UNL, again, Dr. Gary Sullivan, who's sitting right there, they seared the meat in this case, applied HPP, and when they made the cut, the inside still remained red within. So again, because it was not exposed to the oxygen. Yeah, again, related to the exposure of the iron atom within the myoglobin, again, you change, kind of expose it, gets in touch with the oxygen, and as you can see over here, Franklin already kind of mentioned very notable color changes immediately after the HPP. But still, again, in terms of nutritional content, the concentration of amino acids and other, it remains unchanged. How can we minimize this perceived color changes? You can always use use of additional ingredients, marinades. In example, this research study analyzed pet food formulations that were based out of chicken, salmon, and other fruits and vegetables, spices. They added lactic acid and other ingredients provided by Corbion, and we have a representative right here, Rodolfo, who can help you out if you have any further questions. But we'll continuing up with the study. You can see they apply 450 megapascals, will be around 70,000 psi, 3.5 minutes. This amount of lactic acid, 3.6 kilograms or grams per kilogram, sorry. And the log reduction by salmonella, it's low, but the color change, it's not very noticeable. This is due to the added ingredients, the herbs. When you apply higher pressure levels, you can see that it's basically the same appearance. You do have a greater inactivation of salmonella, and even though these conditions are outside of what's applied in the commercial scenarios, you can see that the study also reports that a regular pressure level or 87,000 psi, 600 megapascal with this holding time and lactic acid concentration can do the trick to meet or exceed the salmonella reduction. So again, you can see on the photo that the color change is minimal. The same thing, it's over here reported with the numbers, the lightness or the opaqueness. Before and after the HPP, it's a smaller change. Perhaps in terms of the redness, again, it remains over the same value. So again, ways that you can improve. This is also been done by some of our customers. This All Neighborhood Food, they do the marinades and apply this for different plant for different proteins derived from beef, chicken, pork, and many more. Parameters that influence pathogen inactivation: the greater the pressure level, the greater the inactivation. Particularly for the raw diets, you will basically be jumping directly to the highest pressure level, but if you combine with other hurdles, again, some of the organic acids or salts derived from the organic acids, you might be able to lower the pressure level, and that's part of the research projects carried out here in Nebraska University. Holding time, well, the longer you're pushing, the greater the inactivation. Machines can just go forever, make a cycle of about an hour, but after, if you exceed a 5-minute holding time, perhaps it's time to go back into the kitchen and reformulate. Remember that HPP is batch processing. You want to maximize throughput, optimize conditions, and again, you want to keep it short. Perhaps a gentleman here from Universal Pure will be touching more on the economics later on this. In terms of the food formulation, well, the pH, as in any other intervention, the more acid the media, the more inhospitable environment for pathogens or microorganisms in general to grow. Water activity, Zach will be talking more about this later, but for HPP, you need high water activity values, 0.96, 97 or greater, in order to maximize reductions. If you're seeking HPP to be your lethal step, you need 0.96 or greater water activity. Below that, you complement either as a post-lethal intervention, like the case of pre-cooked sliced deli meats. Combine, in example, during the elaboration of salami, where fermentation, maturation will take away a couple of log reductions. With HPP, you complement, and both or the whole process will exceed the 5 log reductions of the pathogens, or just apply it as a shelf life, again, for foods with low water activity values. There's plenty of information over there reported in inactivation of microorganisms in raw mussel mainly, but it's way harder to have this data for multi-component ingredients, how these interact. Also, you need to take care of, they have been mentioning, or perhaps they will mention that fat content plays a role and may provide a protective effect on microorganisms. And you can see over here very briefly how the inactivation of salmonella in organ meat from pork, well, it's a little lower when compared to inactivations in the raw mussel. So I'm just wrapping up this section with some of the frequently asked questions really quick, because we can certainly extend the time over here of the presentation. Well, the first point related that you need to apply HPP before freeze drying, and it is associated with least water activity targeting 0.96 or greater if you want your HPP to be your lethal step. Reasons are, well, that high water activity levels, 0.96 or greater. If you imagine the microbial cells as a balloon that's completely full with water, since it's completely full with water, the plastic, or in this case the membrane, is stretched to its maximum capacity. If you hold it in your hand, it just takes a bare squeeze, a slight scratch in order to pop or burst the balloon. So similar thing with microbial inactivation. Lower water activity levels, the cell, by adding sugars, salts, solutes, or by removing water through drying or freeze drying, well, the cell will try to equilibrate. So the cell will try to get some water outside of its surroundings. So the balloon, in this case, it's still full with water, but it's not filled to its maximum capacity. You hold the balloon in your hand and squeeze it, it will wobble. You can still kind of break it apart, but it will certainly take you more effort. And again, the same thing happens here with the dehydrated cells. It takes more effort to make the compression and decompression, and that's why you see lesser inactivation for dried foods. Apply HPP on thawed formulations. You cannot do this for frozen products. So water at high pressure or close to the freezing point, 32 Fahrenheit or so, will actually expand, and you rely on this compression and the changes on the compression, decompression, the structural, the mechanical stress on the cellular membrane to break apart. If the water actually expands, compression is lesser, as in the case of the ice, and that's why you have a lesser inactivation. In terms of the fat content, the greater fat content, the more resistant the microorganisms are. There's a protective effect. One thing to think about it is water and fat don't get along, so They will be separate. The other is, well, water by itself is a polar molecule, implying that it has a positive and negative charge. When you apply pressure and reduce volume, they can get attracted or to reduce the space. So it's more compressible compared to fats, which they do not have any charges, or they have either a positive charge, both of fat molecules. So like magnets, if you pull them apart, you can just place them up to a certain distance before they start repelling each other. Under pressure, again, fats will get compressed to a certain point, but they will not allow any further compression. And again, you rely on this compression, decompression changes on, or to generate mechanical stress on the cellular membrane. Don't expect HPP to basically be your magic wand and get you out of trouble. So it's a full approach, and HPP is just a part of a bigger equation. So the ending portion of the presentation, again, HPP, again, just provides too many advantages in terms of the safety, shelf life extension that processors are seeking, but meeting the demands for minimally processed or fresher foods that are sought by consumers, in this case, pet and pet parents. We saw how it's relatively easy to scale up in terms of the processing part. Formulation, well, you always need to go about and tweak, but again, it's easier to implement in pilot scale or greater production when compared to other technologies. And again, it allows you to explore new recipes, to go into new markets.